Hey guys, thanks for joining us on another issue of Try Science, and here we are going to look a little bit more at the characteristics of life by comparing cells to viruses, and then we're going to introduce to you the Cell City Project. So thanks for climbing aboard, and let's check out our first question of the day. Are viruses considered alive? In order to answer that question, you're going to have to know a little bit more about what a virus is and what a virus does. So what I'd like you to do is take out a sheet of paper, make a Venn diagram. If you have two big circles that intertwine, let's put virus on the left, cell on the right. And then we're going to put down some traits that we know about viruses. Let's see what we can figure out about viruses.
Viruses are easily the most abundant life form on Earth, if you accept the proposition that they're alive. Try multiplying a billion by a billion, then multiply that by 10 trillion, and that, 10 to the 31st power, is the mind-boggling number of individual viral particles estimated to populate the planet. Viruses come in many shapes and sizes, although they're all small and infect everything, including plants and bacteria. None of them work in precisely the same way. Is a virus a living thing? Maybe, sometimes. It depends on location.
Outside of the cell, a viral particle is inert.
On its own, a virus can't reproduce itself, or, for that matter, produce anything at all. It's the ultimate parasite. Viruses travel light, packing only the baggage they absolutely need to hack into a cell, commandeer its molecular machinery, multiply, and make an escape. A virus's travel kit always includes its genome and a surrounding protein shell, or capsid, which keeps the viral genome safe, helps the virus latch onto cells and wiggle inside, and on occasion, abets its offspring's getaway. Some viruses also wear greasy overcoats called envelopes. They're made from stolen shards of the outer membranes of the last cell they infected. Influenza, hepatitis C, herpes viruses, HIV, and coronaviruses all have envelopes. These greasy overcoats break down in soap, which is why you should wash your hands often. For a virus to spread, it must first find a way into a cell.
Penetrating a cell's perimeter isn't easy.
Yet, viruses have ways of tricking cells into letting them in. Typically, a portion of the viral capsid will have a strong affinity to bind with one or another protein, dotting the surfaces of one or another particular cell type. Viruses use proteins sitting on the cell's surface as docking stations. The binding of the viral capsid with that cell surface protein serves as an admission ticket, easing the virus's invasion of the cell. The viral genome, like ours, is an instruction kit for the production of proteins the virus needs. This genome can be made up of either DNA, as is the case with virtually all other creatures, or its close chemical relative, RNA, which encodes genetic information just as DNA does. Most mammal-infecting viruses' genomes are made of RNA. Another crucial protein or enzyme for viruses is known as a polymerase. Inside the cell, polymerases generate numerous copies of the virus's genes, hijacking the cell's molecular assembly line to produce capsid subunits and other viral proteins. Capsids, a virus's protein shell, self-assemble from their subunits. Freshly made copies of the viral genome are packaged inside newly made capsids for export. Sometimes the escape is violent and involves the new viruses punching through the outer membrane of the host cell, a process known as lysing. But enveloped viruses can escape by an alternative method called budding, whereby they wrap themselves in a piece of membrane from the infected cell, and cloaked in these newly acquired greasy overcoats, slip through the cell's outer membrane. Even then, the cell having birthed myriad baby viruses is often left fatally weakened.
Okay, and here's some more information that you guys can use in your flow chart in your Venn diagram to compare viruses to cells. Go ahead and pause and take a look at the characteristics of a virus compared to a cell. Okay, let's go back and see how we did. There's lots of things we could put on here, but let's talk about some of the basics. We know cells contain organelles, little organs or places to do cellular processes, whereas a virus lacks that, other than the outside covering or membrane capsid or different envelopes that they're called, similar to a cell wall, but they're not going to have the organelles. The cell can complete all of the life processes, whereas a virus needs to infect a living cell to be able to take it over, kind of hijack the cell to complete its life processes. So how are they the same? Well, they're both going to contain genetic information, and this code becomes the key for how the virus tricks our cells. As you saw, the virus DNA or RNA tricks our cells into creating virus components, which are then released to continue the process. But a virus by itself, without a living cell, doesn't do any of the life processes. So using energy, viruses do not do that unless they're within a cell using your energy. Growing and developing, there's no change from this virus once it's made. Responding to the environment, questionable, depending on how you define respond. Reproduction only within a cell. Adapting, I'm going to say yes, because over time, because of genetic changes, they are definitely going to evolve and mutate. And levels of organization, we can say yes, there are different, at least, components of a virus, but they're not using any of the life processes alone. If we say in a cell, they would have all of these things happening, but they're really using your cell. Before we leave viruses, especially now, let's answer this question. To remove viruses from your skin and hands, which is better? We see sanitizer all over the place, and there's the old soap and water. Well, it turns out different viruses respond differently to different solutions. Let's take a look. Which is better?
Your hands up close are anything but smooth. With peaks and valleys, folds and rifts, there are plenty of hiding places for a virus to stick. If you then touch your face, the virus can infect you. But there are two extraordinarily simple ways you can keep that from happening: soap and water and hand sanitizer. So, which is better? The coronavirus that causes COVID-19 is one of many viruses whose protective outer surface is made of a lipid bilayer. These lipids are pin-shaped molecules whose heads are attracted to water and tails are repulsed by it. So, in water-rich environments, lipids naturally form a shell like this, with the heads outside and the tails inside. Their shared reaction to water makes the lipids stick loosely together. This is called the hydrophobic effect. This outer structure helps the molecular machinery of the virus break through cellular membranes and hijack our cells. But it has thousands upon thousands of weak points where the right molecules could pry it apart. And this is where soap comes in. A single drop of any brand of soap contains quadrillions of molecules called amphiphiles, which resemble biological lipids. Their tails, which are similarly repulsed by water, compete for space with the lipids that make up the virus's shell. But they're just different enough to break up the regularity of the virus's membrane, making the whole thing come crashing down. Those amphiphiles then form bubbles of their own around particles including the virus's RNA and proteins. Apply water and you'll wash that whole bubble away. Hand sanitizers work less like a crowbar and more like an earthquake. When you surround a coronavirus with water, the hydrophobic effect gives the bonds within the membrane their strength. That same effect also holds the big proteins that form coronavirus's spikes in place and in the shape that enables them to infect your cells. If you dry the virus out in air, it keeps its stability, but now surround it with a high concentration of an alcohol, like the ethanol or isopropanol found in most hand sanitizers. This makes the hydrophobic effect disappear and gives the molecules room to move around. The overall effect is like removing all of the nails and mortar from a house and then hitting it with an earthquake. The cell's membrane collapses and those spike proteins crumble. In either method, the actual process of destroying the virus happens in just a second or two, but doctors recommend at least 20 seconds of hand washing because of the intricate landscape that is your hand. Soap and sanitizer need to get everywhere, including your palms, fingertips, the outsides of your hands, and between your fingers to protect you properly. And when it comes to a coronavirus outbreak, doctors recommend washing your hands with soap and water whenever possible. Even though both approaches are similarly effective at killing the virus, soap and water has two benefits. First, it washes away any dirt which could otherwise hide virus particles. But more importantly, it's simply easier to fully cover your hands with soap and water for 20 seconds. Of course, hand sanitizer is more convenient to use on the go. In the absence of a sink, use the sanitizer as thoroughly as possible and rub your hands together until they're dry. Unfortunately, there are billions of people who don't have access to clean drinking water, which is a huge problem at any time, but especially during an outbreak. Researchers and aid groups are working to provide solutions for these communities. One example is a device that uses salt, water, and a car battery to make chlorinated water that kills harmful pathogens and is safe for hand washing. So wherever possible, soap and water are recommended for a coronavirus. But does that mean it's best for every viral outbreak? Not necessarily. Many common colds are caused by rhinoviruses that have a geometric protein structure called a capsid instead of a lipid membrane. The capsid doesn't have nearly as many weak points where soap amphiphiles can pry it apart, so it takes longer for soap to be effective. However, some of its surface proteins are still vulnerable to the destabilizing effect of hand sanitizer. In this and similar cases, hand sanitizer may be more effective, especially if you then wash your hands to remove residual particles. The best way to know which to use for any given outbreak is to do what's best for all things illness-related. Follow the advice of accredited medical professionals.
Need help timing 20 seconds of hand washing? Start now. Wow, what nice clean hands you have. Oops, don't touch your face. You look well rested. Have you been stretching? Must be all that water you're drinking. Happy birthday to... I don't think we have the copyright on that. Well, happy birthday... I forget the words. Has it been 20 seconds yet?
Sub-Zero, Coronian. Fight.
So guys, got to keep yourself safe, keep yourself clean, and now you're armed with all the necessary information. Let's take a quick review of what we know about cells. Here's an animal cell, here's a plant cell. Notice they have many parts that they share in common. Let's start up here. Ribosomes making proteins, which are basically the building blocks for cell parts, chemicals that are directed by the DNA. The DNA is found in the nucleus. The nucleus is often surrounded by the ER, endoplasmic reticulum, studded with ribosomes. Information from the DNA, in the form of a messenger RNA, leaves the nucleus and finds its way through the ER to these ribosomes, which tell the ribosomes the code to make a certain protein, or just a cell chemical, basically. Now, those chemicals are used to build cell parts and do other things, such as leave the cell. Now, Golgi body, Golgi apparatus, in both plant and animal cells can make these little capsules containing your protein. They can even fuse with the membrane and send those materials outside. They're kind of like the way we transport things around like we would at a post office. Mitochondria. All of this is going to take energy. Mitochondria are where we break down food using oxygen to create energy, the powerhouse of the cell, if you will. What else do we have? Vacuoles. In animal cells, small. Plant cells, quite large. Animal cells are rigid. They cannot move. Therefore, they'll have a large vacuole to store water. Why are they rigid? They have a cell wall. Outside their membrane, a sturdy, thick cell wall made of cellulose, which actually adds protection and shape to their cell. Membranes are a thin inner coating, only found an animal cell without the wall. And they are permeable, which means some materials can get in and out. And in chloroplasts are only found in the plant cell. They're green because of a pigment called chlorophyll, which absorbs energy from the sun, helping convert carbon dioxide and water into sugars and conveniently releasing oxygen. So that's a review of some of the cell parts. Our cytoplasm is the fluidy part where all these organelles are located. Okay, and there are some others which you can look up that do different parts. I mean, different things. Each cell needs to complete jobs or functions similarly to the organization you might see in a city or a castle. And you can kind of make some really good connections. And that's what this project's all about. Create a map-style display of a city or other choice, like castles or other things that represent cells. You're going to create a visual and written metaphor of your 10 organelles. So what do you need to do? Have some sort of colorful representation of your city, which you decide, your theme. You could use a city, a castle, or a school, or any other kind of large place that has organization levels. And we want to have labels of your structures that are metaphorically filling in for your organelles. To explain what you're doing, you're going to have 10 well-written sentences to describe the metaphors and say how they relate to actual cell parts. So let's take a look at how you're going to be graded. First of all, have your organelles metaphors accurately describing the cell parts. Labels with drawings for your city. And of course, your effort, creativity, neatness, all are included in your project grade. Let's take a little bit of a closer look and do some examples. In your packet or in your Cell City folder in Schoology, you'll find all these documents. For example, chromosome is a cell part that we're going to say is a folder that contains laws or rules located in the mayor's office of your city. Why? Because both chromosomes and folders containing laws protect the important information and directions in that location. Now, in the cell, that would be the DNA, and in a city, it would be a document that perhaps told us rules or laws of the city. Okay? For each organelle, you will complete a metaphor such as this. We have a place for you to do a rough draft in Schoology where you can do it online. That is a required task that you need to complete so that you can get feedback and credit. The nucleus is the what of your city and why. Okay, I'm going to give you some more examples to look at when we talk about Cell City.
Cells are made up of organelles, which are cellular structures of many different shapes that serve a specific purpose. Think of them as pieces of a city. Organelles keep the city running smoothly. This is me, the nucleus, otherwise known as the mayor of the cell. I store cell DNA, direct other organelles, and contain the information the cell needs to do its job. Here's City Hall, where I work. As the mayor, I spend my days inside making laws and telling the other organelles what to do. Next to the nucleus is the endoplasmic reticulum, or ER, also known as the highway system of the cell. There is a rough ER, which has ribosomes attached, and a smooth ER, which does not. The rough ER produces proteins. These proteins are transported to the Golgi apparatus with vesicles, or a small sac. The smooth ER produces lipids that are used to build cell membranes. As our vast highway system, the ER manufactures proteins that are transported by the vesicle delivery trucks.
Great. So you get the idea, hopefully, and that video can be found right in your Cell City folder. And you can take a look at more organelles to get some examples. But be unique and creative. Make it your own. When you're finally ready to do your final draft, I've given you a document that you can type in, and you can complete your metaphors in a well-written, refined, revised way so that they are completely accurate and make a lot of sense. So guys, let's get working on that. Your rough draft will be due next week, and then your final copy will be due soon after. So rough draft March 5th, okay? Hey, thanks so much for trying science. If you've got any questions, check out the resources on Schoology. Make sure you're keeping up on all the things. And I'll see you on the next one. Bye for now.